Hello, welcome everyone to our virtual lecture series today. It's a great pleasure to have Mark Hartmann with us here today to present his recent work. But before we start with this, let me introduce our next talk, which will be on September 6th by Céline Ballot. We're really looking forward to that as well. But today we first start with Mark, some of the housekeeping notes. Mark will give a presentation of about 30 minutes, which is then followed by a 15-minute discussion. And you're welcome to raise your hand at the end of the talk if you want to ask a question, or you can also post it in the chat and then we will read the question. Maybe a few words about Mark before we start. Mark studied biochemistry in Frankfurt and then did his doctorate in Heidelberg in a joint project with two groups from DKFZ and the University of Heidelberg. And his focus was on basic epigenetic research, chromatin regulation, and epitranscriptomics. And then he moved on for a postdoc in Heidelberg as well at the DKFZ at the section of translational cancer epigenomics, the group of Daniel Lipka. And he has a strong interest in cancer epigenomics, personalized oncology, multi-omics, single-cell analysis in wet and dry lab, and also a strong interest in cellular origins and fetal-like signatures in pediatric leukemias. We are very much looking forward to your talk. I have an expert here today on epigenomics. So welcome, and the stage is yours.
Yeah, Julia, thank you very much for the kind introduction. I'm super happy that I can present our data today. As I just said to Jörn Walter, I presented this project at the very beginning in one of the SCOG meetings at Saarland, and I'm happy that we moved forward and we are quite in a mature state now writing the manuscript. And today I want to hear your opinion about Onco-Fetal reprogramming in a pediatric entity. So let me introduce you to our system first of all. So in the Lipka lab, one of our main interests is the role of epigenetics in normal and malignant hematopoiesis with a focus on myeloid malignancies. And juvenile myelomonocytic leukemia is such a disease which is characterized by proliferation of granulocytes and monocytes. And importantly, this rare disease affects sometimes very young children in an age span of about 0 to 6 years. And this unique neoplasm is characterized by quite simple genetics with a single mutually exclusive mutation of one of these factors here, which leads to constitutively active RAS signaling. And only few secondary mutations are known in a subset of patients only. And in contrast to such very simple genetics, clinical profiles are highly heterogeneous with rare cases of spontaneous remission and high relapse rates even after hematopoietic stem cell transplantation, which is the only curative treatment option for high-risk patients. And at the moment, it remains quite elusive what kind of factors determine this heterogeneity. And obviously, as I just explained, genetics alone cannot explain the full extent of heterogeneity that we are observing, and this is why epigenetics has been previously suggested to be of relevance in JMML. And indeed, we and others were able to describe epigenomic subgroups, which I will call epitypes from now on, which are characterized by different extents of DNA hypermethylation. And interestingly, the lowly methylated LM group in blue has the best, and the highly hypermethylated HM group in red has the worst prognosis when we look at this Kaplan-Meier plot here. So DNA methylation subgroups turned out to be significant solitary prognostic biomarkers, which was translated into a clinical routine in Europe and US. And my PI, Daniel Lipka, and my colleague Maximilian Schönung were significantly driving this forward to translate this into clinics and to improve risk stratification in this disease. However, the functional role of DNA methylation in leukemogenesis beyond its role as a prognostic biomarker remains quite elusive. And therefore, we've been aiming at first of all, identifying the cellular origins in JML, and second, at characterizing the associated molecular aberrations. And like this, we hope that we are able to identify novel diagnostic as well as therapeutic factors which are of clinical relevance. When we have from the patients, which is enriched mostly for granulocytes and monocytes, it became obvious that a couple of known stem cell markers are differentially expressed across epitypes. And of note, this also affects factors that have been implicated in different developmental stages, such as, for example, Lin28B as a prenatal HSC marker and AVP that has been described to be a postnatal HSC marker. And this was a bit surprising because we are looking at, as I said, material of mostly mature myeloid cells, suggesting a strong dysregulation of the such hematopoietic stem cell factors, which can be found at the top of the continuous hematopoietic differentiation hierarchy. And for this reason, we decided to choose a cohort of patients that spans the entire spectrum of epitypes as we described them before for this disease for further analysis, and as exemplified here by, for example, average methylation values of genome-wide CpG islands. And in the heat map, you can see the methylation values of 124 CpGs of our JML DNA methylation classifier that has been developed by my colleague Maximilian Schönung. And with those patients and with this material, we applied a multi-modal molecular analysis looking at genotypes, immunophenotypes, transcriptomes, and epigenotypes, and focusing mostly on hematopoietic stem cells, HSCs, analyzing everything in the context of various developmental stages. And we validated our key findings in a novel JML mouse model. And in course of our molecular analysis, we identified promising markers for clinical applications, which we validated in a preclinical mouse model. And today I will mostly focus on the molecular analysis and start with an exploratory single-cell RNA sequencing analysis of mononuclear cells and hence of total hematopoiesis. So we performed 10x genomics of JML and analyzed the data in the context of fetal, neonatal, juvenile, and adult normal references. And what we observed was that JML hematopoiesis contributes to all major lineages, including all major cell types, as you can see in this UMAP on the left. And this confirmed studies that have been published before. And this is also important to be aware of the difference to other leukemias, which are characterized by differentiation blocks where you usually do not find an entire hematopoietic system. And to determine the starting point of molecular aberrations in JML, we computationally extracted single lineages to do something like a pseudotime analysis by a very simple principal component analysis approach. So in this example, you can see in dimension 1 that it reflects the expected differentiation trajectory from HSCs to mature dendritic cells. And in dimension 2, you can see that it reveals a disease-specific component where we see a shift of JML cells compared to the healthy references. And therefore, JML-specific aberrations can be observed already at the beginning of this system, so at the top of the hematopoietic differentiation hierarchy in hematopoietic stem cells. And this can also be confirmed when we look at the erythroid lineage, where pronounced differences are found all over the trajectory again, but also especially in the hematopoietic stem cells. And this is remarkable because we are talking about a myeloproliferative disease, and this confirms that JML stem cells are probably the crucial cell population that we should spend our time with. So this motivated us to dive deeper into stem cell biology of JML, which is why we enriched hematopoietic stem and progenitor cells for single-cell RNA sequencing on one hand, and on the other hand for ultra-low input whole genome bisulfite sequencing. And when we had a look at the typical, quite simple staining of hematopoietic stem and progenitor cells, we observed an extreme large degree of heterogeneity on an immunophenotypic level. So when we looked at CD34 positive, 38 negative HSBCs and stained this further for 45 RA and 90, we typically find hematopoietic stem cells in Q1 in this quadrant upper left. And then, as you can see, compared to the references, the immunophenotypes seem virtually like a fingerprint per patient when we look at these stainings in JMML. So this is why we speculated that we might find quite pronounced epigenomic differences as we observe them on an immunophenotypic level across those quadrants and not only across those patients, since DNA methylation patterns have been described in the context of cell type identities. When we applied a previously published DNA methylation-based cell type classifier by the lab of Christoph Bock, we made a quite surprising observation. So irrespective of the patient, irrespective of the epitype, as reflected by the colors, and irrespective of the immunophenotypic quadrant, as I just explained to you, this high heterogeneity, we observe for each and every sample that we look at on a methylation level by this classifier a very clear and unambiguous HSC signature. So hence the DNA methylomes of CD34 positive JML cells bear clear HSC signatures in any case, irrespective of the immunophenotype. In contrast to this, again, a simple PCA analysis, now looking at the 20,000 most variable CpGs, it revealed quite clear differences across the epitypes, since PC1 perfectly separates the beforehand determined epitypes based on bulk DNA methylation classification. And this means that the typical JML epitypes are conserved in JML stem cells, again, on top of the hematopoietic hierarchy. So in conclusion, the DNA methylomes bear information about both the cell type identities and disease-specific aberrations. Next, we wondered if the JML epitypes translate into transcriptional differences. And for this purpose, we looked at high-risk HM versus lower-risk non-HM patients and determined differentially expressed genes very simply. And quite some well-known hematopoietic regulators and leukemia markers came up, including markers which have been implicated in hematopoietic development. For instance, HBG2 encodes for fetal hemoglobin, and CD52 is a maturation factor in prenatal hematopoietic stem cells. And on the other hand, we could observe CRA-HBP and ABP postnatal stem cells, as mentioned, stem cell markers as mentioned before, which are enriched especially in the lower risk patients, just as we saw it at the very beginning in the bulk RNA sequencing data. And this is an important observation when we think about the sometimes very young age of the patients with a maximum of about 6 years, and where we have some cases of JML that occurs just after birth. And this is why we wanted to analyze JML stem cells in the context of healthy HSC references, namely, again, fetal, neonatal, juvenile, and adult references. And first of all, on a gene level, we could confirm that CRA-HBP is indeed in this data set a postnatal HSC marker, which is especially expressed in low-risk patients or in HSCs of low-risk patients, and vice versa. HMGA2 is a prenatal marker, which is mostly expressed in the higher-risk patients. To further elaborate on this, we calculated global similarity scores using logistic regression, as previously published by Sembejati, and compared JMML HSCs in the columns to reference HSCs of the 4 developmental stages. And in this analysis, everything below 0.5 in gray means dissimilar, and above 0.5 in pink means similar. And I focus the analysis here on JML and reference stem cells only, but of course, we looked for similarities across the hematopoietic system. So we observed that all JML stem cells, irrespective of the epitype, were highly similar to neonatal HSCs. And in contrast to this, to this pan-JML phenotype, higher risk stem cells were more similar to prenatal than to postnatal references. And this was the opposite for the low-risk stem cells, confirming the inverse pattern that I just explained to you on the gene level. However, when you have a closer look at the similarity values per patient, so per column, so to say, it becomes clear that there are positive similarity values across developmental references per patient, rather than that we find a single unambiguous nearest normal reference HSC. And this is why we were aiming at getting a more detailed picture about gene expression patterns in JML stem cells with respect to those developmental stage references. And for this purpose, we calculated the top signature genes per developmental stage in a one versus the rest manner, and could indeed identify more or less specific marker genes per developmental stage, which were never absolutely specific, but strongly upregulated in a particular stage, as you can see here in this seq map. And when we looked at the transcription levels in JML stem cells, it became obvious that those cells are characterized by something like a mixed or variegated developmental signature, as you might call it, including genes that are strongly up or downregulated in red or green, and also where we find genes that seem to be expressed on a more or less physiological level when compared to the healthy references. So typically in pediatric cancers, it has been described that driver mutations can be present already at birth. And this is thought to lead to a maturation block and in consequence to a preservation of fetal signatures. And this is in line with a recent publication that observed that driving RAS mutations in neonatal blood spots can be present of individuals that develop JML later on in life. But this was true only for a subset of patients. So as you can see that especially the higher risk patients seem to be sometimes negative in those neonatal blood spots for RAS mutations. But of course, this is a quite small cohort, and such negative results might technically be explained by small clone sizes or detection limits. However, there might also be an alternative explanation inspired from adult cancer entities. So a postnatal RAS mutation might occur, and that leads to the dysregulation of gene expression programs and in consequence to the reacquisition of fetal signatures. And this phenomenon, known as Onco-Fetal reprogramming, has been described for adult entities and might explain the rather variegated expression signatures that I showed you just before that we observe in the JMML stem cells. However, to the best of my knowledge, Onco-Fetal reprogramming has not yet been described for pediatric cancers. And this is why we moved back to DNA methylomes as a more robust, a more stable molecular signature. And we wanted to look at JMML stem cells again, now as we did it for the transcriptional analysis in the context of normal HSC development. So we've been looking for regions that change their methylation in the course of normal HSC development and identified quite some regions that are depicted here in the heat map that get hypo or hypermethylated from fetal to adult HSCs. And when we've been looking at the methylation values of healthy and malignant stem cells in exactly those regions, we were able to calculate a phylogenetic tree based on the distances between methylation levels across those stem cells. And surprisingly, when thinking about the mixed transcriptional signatures, the DNA methylomes of JMML stem cells revealed a quite clear picture, very high similarities to postnatal HSCs, irrespective of the JMML epitype or risk group. So a different picture than we observed it on the transcription level. So this suggested that JMML stem cells matured to a postnatal stage across birth, at least on the methylome level. To further confirm this observation, we conducted a complementary analysis looking at so-called epigenetic scars, which are methylation signatures of particular steps in healthy HSC development. So we defined epigenetic scars as regions that change the methylation state in one particular step of development and remain stable afterwards, to be able to look at a robust epigenetic sign of maturation. And this definition includes hyper and hypomethylations as depicted here in the schematic. By looking at the step from fetal to postnatal stages, we've been able to identify such scars as reflected in this heat map, and the reference HSCs are annotated in black, where you can find the fetal HSC methylomes on the left here, and the postnatal HSC methylomes on the right. And as you can see, is that clustering confirmed that all JMML stem cells from all epitypes were similar to postnatal references, quite different to the fetal references, confirming what we observed in the phylogenetic tree analysis. And interestingly, besides this clear similarity to postnatal references, especially the hypomethylated regions revealed that some high-risk stem cells were a bit more similar to the fetal references when comparing to low-risk stem cells in blue, that we have here and here, as you can see in this heat map here and in this bar plot on the left. And the same was true when we looked at the step from neonatal to post-neonatal stage, where especially the higher risk stem cells in red seemed to maybe reacquire parts of the fetal signature, but this reacquisition, of course, is a speculation of mine. And this is also reflected here in the bar plot on the left. But most importantly, again, we observed a very clear clustering of all JMML stem cells with the postnatal rather than with the prenatal signatures. So in conclusion to this part here, we observed a discrepancy between transcriptomes and methylomes, where the high-risk JMML stem cell transcriptomes were shifted towards the fetal and the methylomes of the stem cells towards the postnatal stage. So even if the methylome suggested a postnatal maturation state at the time point of diagnosis, with the human material in hand, we cannot finally distinguish between a fetal origin and an associated maturation block and an onco-fetal reprogramming of a postnatal cell of origin. So this is why we were thinking about a system that we can control in a time-wise manner. So to evaluate the hypothesis of onco-fetal reprogramming. And for this purpose, my colleague Maximilian Schönung established a highly precise mouse model for JMML. And what he did was a postnatal induction of PTPN11E76K, which is the most prevalent mutation in human JML. And he induced this not only postnatally, but specifically in hematopoietic stem cells, which is important with respect to what I showed you before in our molecular analysis in the human system. And this led to a highly precise mimic of human JMML, as exemplified here simply by splenomegaly, and as we can also see by reduced leukemia-free survival of the mutant compared to the control mice. But what we've been interested in was mostly the molecular signatures that we find. So again, with this material or with these mice, we performed single-cell RNA sequencing and looked specifically at hematopoietic stem cells in mutant versus wild-type mice. And what we did was a simple gene set enrichment analysis with fetal and adult HSC signatures that have been published before by Li et al. In 2020. And strikingly, we could see that the RAS-activated stem cells revealed an enrichment of the fetal HSC signature and the control HSCs of the adult signatures. And furthermore, when we looked at our human stage-specific signature genes of normal HSC development, which I showed you before on the heat map, we confirmed the very same pattern or a very similar pattern that RAS-activated stem cells showed an enrichment of early and control stem cells of late HSC signatures. And this result confirmed that Onco-Fetal reprogramming on a transcriptional level can occur upon RAS activation, at least in this murine JMML model. And in course of our analysis, we identified quite some interesting factors with respect to potential clinical applications. And we've been wondering if such Onco-Fetal factors are suitable for clinical interventions. So to choose candidates for preclinical drug testing, we were looking for factors which expression was linked to methylation, since we found that methylation is a very early event in JMML leukemogenesis. And second, we were looking for factors that have been described to play a functional role in prenatal HSCs. And third, a very pragmatic reason, we were looking for factors for which a drug exists that we can apply to a preclinical JMML mouse model. And among a couple of interesting genes were a lot of surface markers, and one of those surface markers was CD52, which was indeed slightly upregulated in fetal compared to the postnatal HSCs. But moreover, it was pan-JML upregulated, and especially the high-risk HSCs were strongly upregulated for CD52. And that's exactly the group of patients where we lack suitable treatment options. As mentioned in the introduction, there's only hematopoietic stem cell transplantation, and we have relapse rates above 50% post-HSCT. And importantly, when we think of therapeutic intervention of a surface marker, this holds also true on a surface protein level, which we could confirm via FACS. So for CD52, a monoclonal therapeutic anti-CD52 antibody exists, which is called alemtuzumab. And our collaborators in Freiburg, namely Giovanna Rajak from Miriam Erlacher's lab, applied this drug to JMML xenograft mice, and we observed a certain degree of specificity when we look at CD52 positive human hematopoietic cells that got depleted upon alemtuzumab treatment. And looking at various tissues where we can see engraftment of hematopoietic cells, it became clear that indeed alemtuzumab was able to deplete human CD45 positive JMML cells. And importantly, this also included CD34 positive, 38 negative cells, so hematopoietic stem and progenitor cells, which is the cellular population of interest after all our molecular analysis. So presumably, the most important population can be targeted by this treatment, and we speculated if indeed disease propagation can be disrupted with an alemtuzumab with an anti-CD52 treatment. To formally test this, whether disease propagation could be disrupted by this treatment, we transplanted material from treated and untreated mice into secondary recipients and observed, this was striking, that the engraftment potential of human leukemia cells was virtually gone when we look at this bar plot here. Moreover, this treatment appears to improve leukemia-free survival of secondary recipients of treated mice. So indeed, this anti-CD52 treatment seems to be not only of high specificity, but also of high efficacy. And this brings me already to my summary. So we observed quite pronounced differences between transcriptional and epigenomic similarities to different developmental stages. And this led us to the hypothesis of Onco-Fetal reprogramming in this pediatric leukemia, which could be confirmed in a JMML mouse model. And finally, we identified a couple of novel prognostic HSC-specific biomarkers, of which I showed you CD52, which is in parallel also a suitable drug target in a JMML PDX model, where an anti-CD52 treatment leads to the efficient disruption of disease propagation. And with this, I come to the end, to the acknowledgement. As you can see, many people helped in this project. Of course, I want to mention first of all Daniel Lipka, my PI, and Maximilian Schönung, who I mentioned a couple of times, who is working for many years with me together in the field of JML and epigenetic regulation. Our division head Stefan Fröhling, Claudia Scholl, and then our clinical collaborators in Freiburg, namely Charlotte Niemeyer, Christian Flote, Miriam Erlacher, and of course Johanna Rajak from the Erlacher lab, who did the xenograft mouse work. And then many people contributed with healthy reference samples and or bioinformatic consultants, analysis, and so on, and a lot of scientific input. So thanks to everybody on this list, and thanks to you for listening. And I'm curious to hear what you think about Onco-Fetal reprogramming in a pediatric entity. So thank you very much.
Thank you so much, Mark, for this fantastic talk, for the very sophisticated analysis and the beautiful data you showed. It's really a great example of how combining analyses in human on different levels and then also taking this into mouse can help us to develop hypotheses, but also confirm them and look at them from different angles. So very, very impressive. Thank you very much.
Thank you.
So then the floor is open for questions. Yeah, dear all, please raise your hands if you want to ask something, or write your questions in the chat.
Is there one in the chat? I cannot see it, actually. I cannot open it. Or there is.
There are no questions in the chat yet. Maybe I can start then with a question. I was going really back to the initial motivation you had about the routine diagnostics and where that stands for JMML. Can you elucidate again how you think that your work really will change or adapt the diagnostic routine for this disease?
Yeah, actually, indeed, it is already implemented in EVOC MDS, which is a large European consortium focusing on JML, for example, and it's already the case that if there's a new JML case in course of this consortium, that samples are sent to Heidelberg, and Maximilian Schönung, who I mentioned before, then indeed does the diagnostics. So he established a pipeline where he can quickly analyze, based on DNA methylation array data, the epigenetic subtype, and this is considered by the clinicians in their decisions. So this is one important part. So classically, genetic mutations have been taken, and this is something that we can also see to some part, that, for example, PTPN mutations are enriched in the high-risk epigenetic subgroup as we define them. But this is not mutually exclusive. So there is somehow an association, but we also find PTPN mutations in the low-risk group. So this is something, yeah, that is sometimes hard to understand. Secondary mutations might play a role, for example, but as Maxi could show, is that indeed DNA methylation is a solitary prognostic significant biomarker, and it is used. And now we are aiming at identifying factors such as CD52, as I showed it, because this is correlated to the epitype, as I showed it, and this can be used as a prognostic biomarker in a much more simple way. So you do not need to make a DNA methylation array analysis. You can, in theory, simply use FACS. This is at the beginning now, so we need to validate this further with some of patients, of course. But in theory, then a simple FACS analysis could be done to stratify the patients, at least to get a very quick, cheap and easy first impression of the risk group. Yeah.
Sounds great. Okay, now we actually have a question in the chat from Tim Trish. He's saying, Phenomenal talk. I especially like the demonstration that a blueprint calibrated classifier can be misleading. It is clear what the biological mechanism for the risk differences between the high risk and lower risk epitypes is.
Yeah, first of all, I would not say that the classifier is misleading. So what we clearly observe, and actually I think this is some beauty in this data, is that we have different levels or different components in the DNA methylomes of such cells. So these are different regions. So when we apply the classifier, we get this HSC signature. And actually, we can confirm that in this immunophenotypically defined compartment, there are the stem cells. This we can confirm on transcriptional level, for example. But we have these regions in genome-wide in the methylome that obviously have this HSC signature. This is one part. And the other part is that a way or a part of these cell type identifiers in the methylomes. We obviously also have regions that are disease-specifically dysregulated on a methylation level. And this is the second level. And also always when I think Christoph Bock said this also in a SCOG meeting in Munich some years ago, like the epigenome bears signs of the history of a cell, of the presence, and of the future. And actually, this is what we see, because what I just explained in terms of prognosis, so taking our disease-specific aberrations as prognostic biomarker is a view into the future. Yeah, and we can, with a classifier, we can see what they are at the time point of lysis. They are HSC-like, and with our associations to the different developmental stages, we can also look back into the past where those cells come from. It's because we see these clear signs of postnatal maturation state. So probably they matured in the past to this postnatal developmental stage. So I think this is not misleading per se, this classifier. This simply gives you a different level of information.
Before we come to Jörn, who raised his hand, Tim had a follow-up question saying, related, how have you or other groups performed metabolic analyses, some of all epitype patient samples?
Metabolic analysis.
Metabolomic.
Metabolomic analysis we did not really do with JMML. I'm thinking if I was leading you to this question, or is this an autocorrection and methylation analysis? Because metabolomic analysis we did not really do.
Okay, and then Tim is saying, last, thanks for citing Astrid Behner's marvelous work. Note also her observation that, as in mice and fish, NUP98 NSD1 fusions without sufficient RAS activation produce paradoxically a JMML-like phenotype in pediatric patients, as opposed to the MDS-like phenotype in mouse models. Fascinating disease model, RAS shifting from glycolytic to OXPHOS. So I think that was more a comment from Tim. Thank you for that. But then let's move on to Jörn with his question.
I was really intrigued by the story. I mean, it's a tremendous work, and it really shows how important it is to look into various modalities and use different modalities for intelligent search of potential markers, and also the history of this disease. I mean, this is a fabulous story. The question that I have is this Onco-Fetal reprogramming or regression, or how you would like to call it. I mean, the mouse model clearly shows that it goes in this direction, but for the human, you see correlation that goes with it, but you have no real proof for that, or?
We do not have proof. I mean, we would need material or information that we do simply not have. Yeah, and this is why at the end we did a scientific approach. What do? We look for a model, yeah, where we can control it. But of course, yeah. In Europe, this is not a default, or this is rarely done, that neonatal blood spots are taken from newborns, right? And this would help us a lot. I mean, we have this very good collaboration as I mentioned, to the clinicians in Freiburg and to the EVOC MDS consortium, which is absolutely great, but this material simply does not exist, at least for us. Yeah, and so of course one question would be that, for example, that we take our patients, we look how strong is the fetal-like signature, for example, on transcriptional level, and then we ask the question if we have RAS already present in the neonatal blood spot or not, but we do not have it. Yeah, so so this is why we needed to come closer to this by a scientific approach and using a model. Yeah, and but of course this cannot be absolutely 100% finally be proven with the material in hands what we have. Yeah, unfortunately.
And then I have a technical question. So what you showed to us as differential methylation was based on whole genome bisulfite sequencing.
Yes.
And
yes.
these were just tiles that you looked into and summed up methylation values in these tiles.
Yeah, we did different approaches. So actually, since these cells are so rare, the stem cells, we needed to find a method that is able to produce good data with very few material. And then what we simply did is we applied the single cell bisulfite protocol from Wolfreich to very small bulks. And this was giving us very good quality whole genome bisulfite sequencing data. And then we did different analysis. So for the phylogenetic tree, it's exactly what you said, Jörn. So we separated the genome into, I don't know how many kb parts. And then we looked at the methylation values of all those parts, genome-wide. And then we looked what is associated with the known trajectory in a healthy HSC development from fetal to adults. Yeah, and then we looked at those regions in JML and looked for distances. And this is the one thing. And the other thing for the epigenetic scars, so the complementary analysis, we did indeed in DMR calling, classically with DSS. And then we looked, and then as a second step, we filtered those DMRs as such that they remain stable afterwards, after this one step that we've been looking at. So we have a DMR in one particular step, for example, from fetal to neonatal. And then the DNA methylation value of those regions is supposed to stay stable, simply looking at standard deviation at the end. Yeah, and this is then for us a possibility how to identify regions that change the methylation state at one point very strongly, but afterwards remain very stable, at least in the normal development. And this we use as scars. Yeah. But this can also be, for example, confirmed by a simple principal component analysis, obviously the most important analysis in this project on different levels. So again, when we do PCA with the healthy reference methylomes and the JML methylomes of the stem cells, we find exactly the same pattern again, that the low risk stem cells are very close to the postnatal HSCs. And then there is a component where the high risk HSCs come a bit closer to the fetals, but overall, all those risk groups are far away from the fetal methylation state, which is so different to the transcriptomes. And exactly what you said, it's so important to look at different layers. And for us in a pediatric entity, I would even expand the statement and say it's not only important to look at different molecular levels, but at this young age to look also at different healthy references. Because what we found at the end and what was giving us so much confidence that CD52 is a good factor, a good target, is that it was upregulated against all healthy references. So fetal, neonatal, juvenile, and even adult. So this was a real disease-specific phenomenon that we observed here.
Okay, there are two more questions in the chat. Another one from Tim. He's asking, if you look at the blocks of methylation that characterize normal human tissues in the context of the JMML samples, are some of the blocks being shattered in a way that is informative about mechanism? This seems like such a fundamental model for RAS dysregulated hematopoiesis and perhaps RAS dysregulation in general. For example, if you were to look at blocks versus tiles, is there information loss?
We did not systematically look at this, but what I can spoil with respect to the functional statement here is that, for example, when we look at different chromatin features that have been described by the ENCODE project, when we look at chromatin features, we can see that not all of those features are randomly affected or not affected. So it's really indeed specific chromatin features and mostly bivalent domains. So this makes sense completely in our eyes in the context again of all these developmental programs. Yeah? So it looks like a lot of bivalent enhancers as well as promoters get very often hypermethylated. And this does not reflect the normal methylation state even of the fetals, which one could suggest. It looks like it is a completely new methylation signature that you cannot find in any of the references. So again, very disease specific. And a speculation might be that this is an attempt to maybe work against something that was happening, maybe shutting down again or so, but what we do not know at the moment, unfortunately, but this is of course something that we are interested in, is what is the histone profiles in this disease. This is at the moment a complete black box, but yes, all these ideas that you just traced are of course of interest, but we did not systematically check this.
So much more to do, but before we run out of time, one last question. What did you find from methylation that you didn't find from expression?
Yeah, the clear postnatal signature. On a gene level, or it's hard to say, because actually I did not really spend so much time to look for the differences, to be honest. What we did at the end, what I skipped here to show you was, but I mentioned it, our candidate selection. So we looked rather for similarities or for associations, let's say it like this. So we looked for differentially expressed genes that are associated with methylation differences, as I mentioned it. And this can be one or more DMRs, as we called them, in a certain region. And this gave us confidence, or more confidence, that these factors are of significance, since we saw these pronounced epigenetic differences already at the stem cell level. So this is an early event in the disease. And this is how we looked for similarities. The differences on a gene level, if you mean this, it's hard for me to say. But clearly what I'm still showing here on this slide, I mean, we have quite some pronounced fetal signatures on a transcriptional level, and we have very, very clear postnatal signatures on an epigenetic level, which might lead us to the speculation that one thing comes before the other, but I think this is a dangerous thing, because, for example, we have these fetal-like parts of the methylomes that might be enough to drive it into a fetal transcription program, but we don't know it. And for this, we need to manipulate the system somehow to dive deeper into mechanism of epigenetic regulation, but I can only talk about speculations here then at this moment, so sorry.
Okay, but great. So thank you so much again for this fantastic talk and sharing the news data. It has been a pleasure to have you here. Thanks everyone to listening, and see you at the next meeting in September.